Good evening, everyone. Welcome to YouTube session on Botany Insider. We'll be having a discussion of various topics in the upcoming live sessions on YouTube till your June 2024 examination. We have just received the notification for that. That is, your exam dates for the upcoming examination is now from 25th to 27th of July. So you have exactly 20 days in your hand for you to perform with the best of your ability in the upcoming examination, right? So please make sure that if you are like actually preparing for this examination in the best manner possible, try joining on in these YouTube sessions completely free of cost, and we'll try to cover up as many things as possible in these live classes, right? So make sure to come in the live session on daily basis every day. 8:30 the class will be therein, right? So if you wish, you can come in the live session on daily basis. Good evening, travel sphere. Right now, let's begin with the class. Before starting the video, if you're new to Botany Insider, please make sure to hit the subscribe button. In addition to that, please make sure that we have a number of courses running for you all. One of the course running for the June 2024 batch is the PYQ practice sessions. In addition to that, we have a number of different courses as well. Now, for your December 2024, because the December cycle is not going to change at all. So we should be start preparing. We should start preparing for the December examination also. So for that, we have our full course started. Yesterday itself, we started off with our full course in which all the units will be dealt with. Live and interactive classes for part A, B, and C will be there. Hard copy notes will be delivered to your doorstep. All the recordings will be available. Daily practice questions, PYQ sessions, regular doubt session, weekly tests, and two revision cycles will be there in the entire course. We started this course just yesterday, that is on 1st of July. If any one of you or if any of your colleague is participating in the or start thinking of coming in the December 2024 cycle, please share this particular information with them so that it can be helpful. Right now, very first thing, what are the various things that you should be focusing upon if you're actually and sincerely targeting out your June exam? See, there was a glitch or there was a glimpse of exam getting postponed, some of the other things happening with exam, some exams getting leaked, some exams getting cancelled, some exams getting postponed. But now everything is started to come back on track. You have your examination from 25 to 27th, right? So you have your 20 days in hand. How should you be targeting out these 20 days? I've already uploaded a video, a detailed video for you all today in the morning on our YouTube channel. So if you haven't yet watched it, watch that particular thing. It will help you out. And if I give you a glimpse of that, so very first thing is that you should be focusing upon at least, or you should target at least two more revision cycles in these last 20 days. First thing first. Second thing, try to give as many mock tests as possible. Try to increase your mock test frequency and give more mock tests for your upcoming examination, because the more you practice, the better results you're going to get, right? We have a mock test series also running on our application. So if you want, you can join in that as well. It will help you out. There are more than 120 tests in that entire mock test series. So try joining in that. Then you also have to make sure that you're not skipping your previous year question. So try revising the previous year questions in the best of your ability. Even if you have completed all the mock tests, no worries, but still try to give more mock tests. Try to practice out the previous year question papers in the mock test format, because using those 3 hours in the best manner is also very, very important. So try utilizing or try having a very proper strategy for the 3 hours. Revise your strategy, check out if there was any loopholes, and try getting into the actual state of giving the examination, right? Fourth thing, something that if you feel like you want to add in, do it. Otherwise, just these three things will help you out to like scan through the entire syllabus for your preparation. And the fourth thing is that if you feel like adding some new topics, some new additional topics in your preparation series, try adding on those topics and try to like enhance your knowledge accordingly. Those topics you can take up from your PYQ questions as well, because in the previous year questions we sometimes get such questions where it might be completely off track, it might be completely, or it was not earlier mentioned in your syllabus. So try to work upon that as well. Right now, this is about what should be your target of interest area for these last few days. Now we'll discuss a few questions in today's session because this is a warm-up session. From tomorrow, I'll be taking up a lot of new topics. Like I'm targeting out to take up all the heavy topics of the entire course. For example, cell cycle. For example, your DNA replication is also there. We have the concept of entire cancer topic, right? Then you have the topic of DNA replication, like this is the first one. Then you also have the transcription, translation. All these topics are something that are very important, and I'll be targeting out all these topics in the upcoming live classes, and every day the class will be there at 8:30 only. So try joining in that particular, in this particular session on daily basis. It is going to help you out in the best manner, right? So today let's solve out a few questions and let's get done with the practicing of the same. The first question is, good evening Dimu. The first question is, which one of the following is a major antibody in the early stages of the primary immune response? Question is, which one of the following is a major antibody in the early stages of your primary immune response? You have IgA, IgM, IgD, and IgE. Try to get the answer for this particular question, everyone. It's very basic and it's very simple. So try getting into the answer for this particular question. Everyone mark the answer for this. Which of the following is a major antibody in the early stages of the primary immune response? IgA, IgM, IgD, and IgE. Very good, very good. Others also mark the answer. Try to, like, whatever answer is there in your head, try to write it on in the chat section, and then we'll solve it out accordingly. No worries. Even if you get the answer wrong, it's okay, but try to get the answer for the same. Let's see. Yes. So, the question is, which of the following is the major antibody in the primary immune response? It is your IgM only. Very good. Simple question. The main primary antibody is your IgM. What is Ig in this entire thing? Yes, what is the meaning of Ig over here? Everyone, write down in the chat section. What do you mean by Ig in this entire segment? And over here, remember, it is asking about the primary immune response. That is the first immune response. That's why the answer for this is option number 2. That is IgM. IG stands for, very good, it stands for immunoglobulin. Remember, it is asking about your primary immune response. So you should be reading the question very, very effectively, right? Remember to read the question very properly. Next question, give it a try to this. Which one of the following statement is correct for early embryonic development in terms of differentiation? When you study developmental biology, we go through a number of terminologies like differentiation, determination, specification, induction, potency, etc., etc. So try writing down which of the following is the correct statement for the terminology of differentiation. First statement is saying, good evening, Meenu. Good evening to other students as well. First option is saying the first stage of commitment is termed as specification, while the second stage is determination. Second, if a specified cell is transplanted to a region of differently specified cell, the fate of specified cell remains unchanged. Then, a cell or a tissue is said to be determined if it shows reversible fate under the influence of the surrounding cells or tissues. And last option is the first stage of specification is termed as commitment, where the cell develops autonomously. Question is asking you to define in terms of differentiation. So try marking the answer for the question, everyone. Easy and simple. Try it out. Which of the following statement is correct for early embryonic development in terms of differentiation? We have learned about what differentiation is, what determination is, what commitment is, and all those things. So try marking the answer for this question now. Differentiation. Others also mark it. Very good, very good. Read the question carefully. It is asking you the information which is correct in terms of your embryonic development of, like in the terms of differentiation. First stage of commitment is specification, second stage is determination. Second is saying that if a specified cell is transplanted to a region of differently specified cell, the fate of the specified cell remain unchanged. Then a cell or a tissue is said to be determined when it shows reversible fate under the influence of tissue, surrounding cells or tissue. Then the first stage of specification is termed as commitment, when the cell divide autonomously. Let's look into the answer for the question. Answer for this particular question is option number 1. First stage is specification and the second stage is determination. Question is asking you in terms of differentiation. What do you mean by differentiation? So the process in which a cell actually takes up a particular functionality and behaves accordingly. So all the other things are not explaining you about differentiation at any level. Option number 1 is explaining it to you properly. That is, first stage is termed specification and the second stage is known as determination, right? Remember about this. This is a correct thing to mark. Next question. I hope it's clear. Next question. The researcher, while studying about the vernalization, which is known as a chilling effect or cold treatment, he made following statements and we need to find out the incorrect statement. First one is vernalization causes stable changes in the competency of the meristem to form a inflorescence in some of the plant species. Second one is saying vernalization causes changes in the gene expression that do not involve alteration in the DNA sequence. Third is saying flowering is not altered under normal growth condition in the plants that are defective in vernalization. And last step is vernalization alters the expression of the FLC genes. So Question is asking you that which of the following statement is wrong in terms of your vernalization. Try to mark the answer for the question, everyone. Give it a try to the question. Incorrect statement in terms of your vernalization. Vernalization is chilling treatment or cold treatment. मीनू हर रोज हम लाइव क्लासेस 8:30 to like 40-45 minutes classes will be there on the YouTube every single day till your June examination, जिसके अंदर हम क्वेश्चन सॉल्व करेंगे, हम टॉपिक्स रिवाइज करेंगे और हम साथ साथ क्वेश्चन को कैसे अटेम्प्ट करते हैं एग्जामिनेशन के अंदर, इस चीज को अच्छे से समझेंगे. सो इफ यू वांट, आप डेली बेसिस के ऊपर यू कैन ज्वाइन इन द क्लास एंड अंडरस्टैंड अबाउट द एंटायर थिंग. हर रोज 8:30 सेम टाइम के ऊपर क्लास होगी, राइट? एंड आपको बहुत सारे ग्रुप्स हैं हमारे यूट्यूब के, व्हाट्सएप के ऊपर, टेलीग्राम के ऊपर, जहां पर आपको क्लास के लिंक्स मिल जाएंगे. ओके, एवरीवन मार्क द आंसर फॉर द क्वेश्चन. Which of the following statement related to vernalization is wrong? Very simple question. Everyone mark the answer. 9 of you are there in the class right now. Very good, very good. Everyone read the question carefully. Third option is saying that flowering is not altered under normal growth condition for the plant which is defective in vernalization. Obviously, if the plant is defective in vernalization, it is going to get impacted. Vernalization is what? It is the cold treatment, right? What is it? It is the cold treatment the plant requires to initiate or to induce its flowering. अगर एक प्लांट के वर्नलाइजेशन के फिनोमिना में ही डिफेक्ट है, उसके अंदर ही प्रॉब्लम है, सो प्लांट फ्लावरिंग कैसे करेगा? सो ऑब्वियसली flowering is altered under normal growth conditions in the plants that are defective in your vernalization. So remember this. Third is the correct answer for this question. आपको इस क्वेश्चन के अंदर इनकरेक्ट ऑप्शन देखना था. रिमेंबर, एग्जाम के अंदर क्वेश्चन रीड करते टाइम यू शुड बी फोकसिंग अपॉन कि हमें करेक्ट ऑप्शन को फॉलो करना है या इनकरेक्ट ऑप्शन को, एंड अकॉर्डिंगली हमें आंसर मार्क करना चाहिए. एग्जाम के अंदर मेजॉरिटी ऑफ स्टूडेंट्स ये मिस्टेक करते हैं दैट दे रीड द क्वेश्चन बट दे डू नॉट फोकस अपॉन कि करेक्ट आंसर मार्क करना था या इनकरेक्ट, कि करेक्ट ऑप्शन मार्क करना था या इनकरेक्ट. सो मेक श्योर कि आप इन चीजों का बहुत अच्छे से एग्जाम के अंदर ध्यान रखें एंड अकॉर्डिंगली आंसर्स को मार्क डाउन करें, राइट? आई होप दिस क्वेश्चन इज क्लियर टू ऑल. नेक्स्ट क्वेश्चन, सेम पेपर. This question was also asked. Mobile signal, that is florigen, that controls the flowering status of the plant. It is encoded by which of the following? Try out the question, everyone. The question is that the mobile signal florigen that controls the flowering status of the plants, it is encoded by which of the following? FLC, that is flower locus C. Then you have FD, that is flower locus T. Then you have flower locus T, that is FT. And last one is constans. Try it out. Very good. Very simple question. Everyone mark the answer. Make sure जब आप आंसर मार्क करें तो आप श्योर हो कि हम क्या आंसर मार्क कर रहे हैं. मेन एग्जाम के अंदर आपको 2 बार आंसर मार्क करने का ऑप्शन नहीं मिलेगा. यू विल हैव टू मार्क द आंसर इन द first attempt only. So be very, very sure and very, very attentive जब आप आंसर को मार्क करें. Very simple question. Let's see. Question is, the mobile hormone floragen that controls the flowering status of the plant is encoded by which of the following? So it is your flowering locus T that is helping out in the entire scenario. So C is the correct answer. काफी simple question था. 2 marker. Next question. This is from plant physiology and the question is, phelloderm is derived from which of the following? Try the question, everyone. फेलोडर्म कहाँ से डिराइव होता है? इज इट योर कॉर्क कैम्बियम, fascicular cambium, interfascicular cambium, or the provascular cambium? Try it out, everyone. Phelloderm is derived from which of the following? फेलोडर्म कहाँ से डिराइव होता है? कॉर्क कैम्बियम, fascicular cambium, interfascicular cambium, or your provascular tissue? Try it out, everyone. Very simple question. Everyone mark the answer as well. From where do you derive the phelloderm? Very simple question. So phelloderm is derived from your cork cambium. One is the correct answer for this particular question. Very, very simple, very easy and very direct question. Next question for you all. Before that, we have our December 2024 course, which started yesterday. That is from 1st of July. If any one of your friend or if anyone is uh, targeting out the December 2024 examination, you can let them know about it, that the course has been started and they can enroll accordingly. Right? Then this is a general aptitude question. Try the question, everyone. And also we have reduced the price from this as well. That is 4,999 is the pricing for our full course now, just for the students, so that majority of the students can enroll in our batches. So we have our prices 4,999, right? Give it a try to this question now, everyone. A shopkeeper purchases a product for rupees 100 and then he sells it for with a profit of 10%. Then the customer is reselling the same content, reselling the same uh, product to the shopkeeper back again with a loss of 10%. So in these dealings, how much is the shopkeeper actually making? Or what's the, we can say, what's the final profit for the shopkeeper? Try the question, everyone. Not very difficult. Read the question carefully. Try solving the question before marking it. Just check what the question is asking you. In the second scenario, when the profit is like when the customer is reselling it back, the amount has been changed. Let's see, a shopkeeper purchases a product. Let's say the shopkeeper is purchasing a chocolate for rupees 100. Okay. He is selling this particular chocolate to the customer at a profit of 10%. That is, by increasing 10%, that is increasing 10 rupees, he's selling it to the customer for 110 rupees. The customer is selling it back to the uh, shopkeeper with a loss of 10%. That is, on 110, the customer is having a loss of 10%. That is 11 rupees less. As a result, the shopkeeper gets it back for rupees 99 only. Now, how much is the shopkeeper making in this entire scenario? So, 10 rupees over here and 1 rupee over here. Total 11 rupees will be there, or total 11 rupees will be made by the shopkeeper in this entire scenario. 100, 10% is 10 rupees, and 110 10% is 11 rupees. So, the shopkeeper initially made 10 rupees here and 1 rupee here, because the product which was costed to him for rupees 100, he got it back for 99. That's why answer for this is option number B. That is, he made a profit of entirely rupees 11 and not rupee 1 or no profit, no loss thing. You have to understand what the question is asking you. Is it clear to all now? If the examiner would have asked that what was the loss to the shopkeep to the customer, then the answer would have been 1 rupee, or then the answer would have been something else and not this thing, right? It would have been, the answer would have been rupees 11, right? Or if the examiner would have asked you about that what was the loss or what was the profit of the uh, shopkeeper in the second dealing, then the answer would have been 1 rupee. So, question can be framed in a number of ways. You just have to understand the manner in which the question is getting asked. Is it clear to all? Many of you are there in the class right now. Just check whether the question is clear to you or not. It's an easy question. Second is the correct answer for this question. Is the question clear to everyone? Yes. Is the question clear to everyone? So this is about for today's segment. We'll discuss the remaining question in tomorrow's class. We have our December 2024 batch started yesterday, that is on 1st of July. If any one of you who's targeting out, or if any one of you who is planning to give the December examination, you can enroll in this particular course. The entire course is all in one, and it is something that will help you out to sail through this examination in the best, smartest manner possible. So if any one of you who is targeting out the December examination, you're most welcome in this course. So try joining in the course and get yourself enrolled. In addition, we have mock test series, PYQ practice course for the upcoming examination also, for the June exam also. So whatever is comfortable to you people, you can join in accordingly. Let's meet tomorrow, same timing, 8:30 p.m. Be there in the class and we'll discuss further things in the entire segment. Bye everyone.